Hello classmate, my name is Lee Ji-hyun and I'm in the plant immunology lab. I'm going to talk about what I'm learning in the lab. At first, I'm going to tell you why plant immunology is needed and why they are competitive in the future market, and then I'm going to tell you what is plant immunology, and finally, what kind of skills I'm using in the lab. First, I will tell you why I study plant immunology. Look at the picture on the left. A train is trying to transport food. This train looks like it's hard to keep up with the rapid population growth. Statistics predict that the population by 2050 will reach about 10 billion. But as you can see from the picture on the right, food production on the Earth is hard to keep up with this population growth. Eventually, there will be food shortage. And so far, many people argue that food is a distribution problem because on one side people die of obesity and on the other side they die of hunger. Ironically, even under this situation, some people are trying to make biofuels with foods such as corn. Food shortage seems to be a national problem aimed growing population, poorly distributed, and continuing attempts to make biofuels. To solve this problem, we need to increase food production. Statistics show that 60% more food production can solve this problem. Among the many ways to increase food production, our laboratory's concern is to reduce losses in the food it produces. Food loss from pathogen and insect account for 1/5 of total output, and in countries like developing countries, about 50% of total loss is due to pathogen. So, what are some ways to overcome this problem? The triangle on the left is called the disease triangle, which depicts the relationship of the factors that affect the outbreak of plant disease. Our laboratory is looking to study the relationship between pathogen and plant immunity. Second, let me tell you what plant immunity is. The elements that cause disease in plants can be largely divided into abiotic stress and biotic stress. Here are some examples of that. So, how do plants protect themselves from all these different elements? There are structure and biological defense. Here are some examples of that. Among them, I'm studying the innate immune defense of plants that protect themselves with their own immunity as pathogens enter. Let me explain the innate immune response of plant. First, when pathogens break into plant from the outside, and plants use their pattern recognition receptor to recognize the preserved pattern of pathogen, we call these PAMs. And then plants transmit plant signal, and finally plant immune response happen, like the HR reaction. This called the pattern triggered immunity or PTI. But in order to overcome this reaction, pathogens are also able to evolve a weapon called an effector and inject into the plant. This effector has some effect on the protein of plants so that plants do not recognize pathogens. We call this is effector-triggered susceptibility or ETS. To overcome this, plants make resistance protein that recognize the effector directly or indirectly and protect themselves. This called effector-triggered immunity or ETI, and it is stronger and lasts longer than PTI. Next, I'm going to talk about how to study this plant immunology. So, before I tell you, this is one of the projects that I'm working on. Viral A is an effector of human pathogen called Shigella that invades the human body and cause disease, and is known to combine directly with autophagy to prevent ER to Golgi trafficking and recycling, and interact with a protein called calpain to cause necrosis or apoptosis. There's a study showed that viral A causes disease in real plants, and I'm studying how viral A interacts with the plant protein that causes the reaction. As a way of studying this, the first is histohybrid. This is a method of dividing protein that binds to carbon gene by binding site domain and activation site domain, and fusion your interesting protein each and then expressing it inside the yeast. If they are strongly expressed, the reporter gene that exists behind the carbon binding site will be strongly expressed, which will allow them to produce amino acids essential for yeast or exgal so they can grow on medium with specific composition. On the right side things are vector I use. The difficulty of this experiment is that if self-activation of binding site domain take place, it is difficult to accurately measure the strength of the protein that interacts, and this self-activation depends on the gene. In order to find the protein that you are curious about whether interacting or not, you need to do screening. Using the histohybrid from previous slide, you put a binding site domain plasmid into the library that has a large number of activation site domain plasmid, contains some specific gene, and we find the gene that they interact with. This method is to check binding between protein, and this is what I use is to strain, and the difficulty of this experiment is making chance to meet binding site domain and activation site domain. Now we have found that genes interact within the yeast, but whether they interact in the real plant is another question. If you want to know if they are actually expressing and interacting with plants, you have Western blot and co-IP that you can check the amount of protein. But there's also a BIFC that you can see with your eyes. BIFC is bimolecular fluorescence complementation. The principle of BIFC is as follows. Divided YFP into N-term and C-term and connect each target protein to the N-term and C-term and express in plant. You can observe the YFP with a fluorescence microscope, and the stronger the interaction, the stronger the light will be. This is what I use vector in BIFC. This is the process of doing BIFC, which first infiltrated the agro into tobacco and then observed the results 3 days later. The advantage of this experiment is that you can visualize the interaction of protein. However, the downside is that the signal can be arbitrarily emphasized to control its intensity, so you should be careful about it. This is what I prepare for our seminar class. Thank you for listening.